Okay, can everybody hear me? Yes. Good morning, everybody. Thanks for joining us today. My name's Catherine, and I am the CEO of the Baird Institute, for those who've never joined one of these webinars before. I want to welcome you, as I said, and thanks for joining us today. Thank you also for your support of the Baird Institute, whether you're a first-time donor or a long-standing donor. Thank you. Your support really is appreciated and really helps with our research, as you'll see today. The webinar today will explore cutting-edge research into human cardiac ageing, led by experts from the Baird Institute's Centre for Heart Failure and Diseases of the Aorta. As our global population ages, understanding how the heart and aorta change over time is critical to tackling major conditions like heart attack, heart failure, and aortic aneurysms. Using unique samples from the Sydney Heart Bank, the team is building the first comprehensive molecular map of cardiac ageing. Ultimately, this work aims to unlock new therapies to prevent and even reverse age-related heart disease. I'd like to introduce you to some of our team who are presenting today: Professor Paul Bannon, Professor Sean Lal, and Dr. Cassandra Malecki. Paul is the chair of the Institute, of the Baird Institute, which was established in 2001 to improve outcomes for heart and lung surgery patients. He is the head of research for the Department of Cardiothoracic Surgery at Royal Prince Alfred Hospital and holds the professorial chair of cardiothoracic surgery at the University of Sydney. In addition, he is a co-director of the Centre for Heart Failure and Diseases of the Aorta. Paul has performed over 5,000 cardiac surgeries, from bypasses to complex aortic arch and root reconstructions. Sean is a professor of clinical and molecular cardiology at the University of Sydney. He's also a consultant cardiologist specialising in advanced heart failure in the Department of Cardiology at Royal Prince Alfred Hospital. He's the director of the Sydney Heart Bank, which underpins several cardiovascular research programs nationally and internationally. Sean is a group leader in the School of Medical Sciences at the University of Sydney and co-director of the Centre for Heart Failure and Diseases of the Aorta, a partnership formed by the Baird Institute, the University of Sydney and Royal Prince Alfred Hospital. Finally, Cassandra is working as a postdoctoral fellow of the Baird Institute and the University of Sydney and is the biobank manager of the Sydney Heart Bank. Her current research focus is on utilising the precious resources of the biobank to understand mechanisms of cardiovascular ageing, aortic disease and heart failure. Cass completed her PhD in the aortic research group under Associate Professor Brett Hambley and Professor Richmond Jeremy at the University of Sydney. Just before I hand over to the team to do their presentation, a little bit of housekeeping. All attendees' microphones have been muted, just to be sure that the webinar goes smoothly. But if you have any questions throughout the presentation, note them down and you'll be given an opportunity to ask at the end of the presentation, or just use the Q&A button that's at the top of the webinar page in the banner at the top to send a question through during the presentation, and we'll answer all questions at the end. So just to let you know, Professor Bannon, I don't think is online at the moment because he has a patient that's just gone longer than normal, but he'll pop in when he can. So Daria, I think maybe you take your screen off and Cass share her screen now. Okay, so over to Sean, I think, to start.
Thank you, Catherine, and thank you everyone for joining. And it's a pleasure for Cassandra and I to give an update regarding two aspects, so both related to the recent appeal as well that you so generously donated to. One about the Sydney Heart Bank itself, which you've heard about before, but just to provide an update with that. And then also specifically about our program, our exciting program around cardiovascular aging. And that really is about aging of the human heart and also aging of the human aorta. And both of these projects have had exciting developments. We've got some interesting experiments planned, and importantly, they are novel and groundbreaking because really they are the first studies to examine how the human heart and how the human aorta change with time. So over the lifespan, how it ages. And of course, that is intricately linked to cardiovascular disease because diseases of both the heart and the aorta, the biggest risk factor is aging and our population is aging. Yet there have been no fundamental studies that have looked at how the human heart and aorta age. And so it is exciting for us to give you this development, and we're very grateful for the research support that we have had. But first, I'd like to talk about the Sydney Heart Bank. And the Sydney Heart Bank is a really unique resource. It really puts Australia on the map in terms of being able to facilitate human heart and aortic research. It is the largest human heart bank in the world, almost at 40,000 samples. It began in 1989. And it's a testament to the University of Sydney that since 1989, they have maintained this amazing resource, which I was fortunate to be a part of when I was a research student with Christos Remedios. And it was started by Christos Remedios, who's now an emeritus professor at the University of Sydney, but also Dr. Victor Chang back in 1989. And the University of Sydney has been solely responsible for maintaining this resource, which is completely not for profit. And historically, it has had these samples taken from patients who underwent heart transplantation at St. Vincent's Hospital in Sydney. And it also included samples or donor hearts that could not be used for heart transplant simply because of logistical reasons, but the hearts themselves were actually normal. And so we've been collecting these samples since 1989. But where the Baird Institute has really been instrumental, and by that virtue through your generous donations, is supporting the Heart Bank in more recent years, and in particular, the collections that we are currently undertaking from Royal Prince Alfred Hospital, samples that relate to hypertrophic cardiomyopathy, samples that relate to valvular disease and coronary artery disease, where we take biopsies from the left ventricle, these little small snippets from the left ventricle during cardiac surgery by someone such as Professor Paul Bannon undertaking that surgery, as well as samples from the aorta from patients who have an enlarged or diseased aorta or have had an aortic dissection. And then even more recently, actually undertaking banking from vascular surgery as well. So it really makes the Sydney Heart Bank a unique resource because we have these historic samples from patients undergoing heart transplantation, as well as these unique donor hearts that are actually healthy and span the lifespan from 3 months old all the way to 65 years of age. And we also then have these samples from Royal Prince Alfred Hospital taken in different disease sets. And so it really is an amazing partnership between the Baird and the University of Sydney in maintaining this truly unique resource that has a global mission to facilitate research into heart failure, aortic diseases, and cardiovascular aging. And at present, we have almost 20 or 25 international research collaborations with institutions such as Oxford University, Harvard University, Stanford, Mayo Clinic, etc. So, top institutions, but more importantly, we are also undertaking research here in Australia at the University of Sydney, Royal Prince Alfred Hospital, and the Baird. And that's what we'll give an insight into. Sandra, if we go to the next slide, please. Thank you. So just to give an update in terms of the current collections, the ones from RPA, we can see that in terms of individual patients, we're now up to sort of almost 70 myomectomy samples from patients with hypertrophic cardiomyopathy. These biopsy samples taken during cardiac surgery, such as valve surgery or coronary artery bypass surgery, around 50, 360 odd aortic samples, and the vascular group is about 80. Now, that's number of patients, but you can appreciate that individual samples from those patients would be more than that. And so we're probably looking at somewhere of the order of, maybe 1,000 or two in terms of actual samples, or even more than that. So it really is a unique resource. And for example, there is no other heart bank in Australia that has those types of samples that have been collected. And you can see why so many international institutions want to collaborate with us. But what we'd like to talk about today is some of our own research. And this actually relates to those historical samples that I mentioned earlier, because these control hearts, these donor hearts, which are otherwise normal when you look at them from a pathology point of view, These samples are really unique because they span from, as I mentioned, 3 months old to 65 years of age. So it gives an opportunity for the first time to understand how the human heart ages. And then uniquely, not to the same extent, but we do also have some normal aortas as well. So these were taken at the same time when these donor hearts were procured. And that gives us an opportunity to look at how does the aorta age. And as I mentioned, both points are really key to understanding why age is the biggest risk factor for cardiovascular disease. Because if you don't understand at a molecular and cellular level how the human heart ages and how the human aorta ages, you then position to understand how does those changes of aging lend itself towards disease. And so that's what we're going to update you with, and some tremendous work that's been led by Cassandra, and I'll hand it over to her.
Thanks, Sean. So Sean said, I'm going to kind of share how we've been able to use these control samples to look at cardiovascular aging. So Catherine touched on it at the beginning, but why aging again is so important is we know the population is rapidly aging. So it's expected by 2054 that the number of people aged 65 years and over will actually hit about 1.6 billion. And this is more than double the figures that were reported in 2024. And so you can imagine, obviously, as the world is aging, so is kind of the burden of age-related diseases. And we know that age is one of the strongest risk factors for developing cardiovascular disease, so including heart failure and aortic disease. And as Sean mentioned, there's really been no research that has a real clear understanding of how these systems, the cardiovascular system, changes with age at a molecular level. Most studies have been done in animals or in very small cohorts. So we're very lucky that we're in the position to use the samples of the Sydney Heart Bank to explore this research question. And so today I'm going to talk about kind of two arms of my research, where we've been one using those control human hearts of the heart bank and looking at how the human heart changes with age. And then the second part of the presentation will go into the findings we've been kind of exploring, looking at the aging of the aorta. So starting off, I'm going to talk about the aging of the human heart. And this was really excitingly published late last year in Aging Cell, which is a really top journal in the aging field. So that was really exciting to get that published. And so what we did, our study design, was using those healthy hearts from the Sydney Heart Bank. We took 12 hearts that we classified as younger. So these are from people that were aged 25 years or less. And we compared these from hearts of older individuals, so 13 hearts of people who were aged 50 years or over. And we did a range of analysis. So we looked at the proteins, metabolites, lipids or fats within the hearts, and how genes were expressed as well. And so we ended up with a massive amount of data and did all these different types of analyses, which led us to a lot of interesting results. But I think one of the key things that came out of this study was the fact that we developed this first molecular resource of human cardiac aging. So it's just a little video of the website that we developed. So once our paper was published, this was made publicly available to all researchers, so they can click on the little URL in the paper. And what it allows for is anyone interested in this field of aging to easily go into the website. They can search, these are just proteins here that someone's looking up. They can look up, for example, a protein of interest and see how that changes with age. They can see all of the analyses in a really clear way. So it really helps anyone to really dig into our data and look at something that they may be really interested in. And so we're hoping that this will allow researchers in the aging field to really move forward in understanding what's happening with the heart with aging. And so from, you can imagine, like I said, a massive amount of data, there are a few things that we were quite interested in that our lab has started to kind of pursue and understand further. And one of them was a change in this protein known as SERCA. And so we saw that there was quite a substantial decrease in these older healthy hearts. And so we saw this at the protein level. So you can see here that the number here is the amount of SERCA. And you can see the blue bar is the younger individuals and the orange bar is the older. And you can see that the amount of SERCA is significantly lower in the older hearts. And so this was a really interesting finding to us because we know that circuit actually decreases in heart failure. So the fact that we're seeing it decreasing in healthy hearts kind of suggests that it actually may have an earlier role to play in just how the heart naturally ages. We then also wanted to kind of validate this. Usually in science, showing it one way is not usually enough. People usually want to see the result in another way. So we also did some really cool imaging. So the picture here, what it's showing is an actual microscopic slide of heart tissue. And so anything in purple is actually staining for that SERCA protein. And you can see that the top square has a brighter purple color than the bottom, showing that there's more SERCA present in a younger heart compared to an older heart. And this is then when we quantify this and graph it, you can see that too with the bar graph. So this was, like I said, a really cool finding. And why this was important in terms of what SERCA is responsible for in the heart is that it's really important for how our heart can relax. And so we know as well as we age that our heart really does start to dysfunction in this respect, in that naturally its ability to relax will not be as efficient, and our hearts do become stiffer even with naturally aging. And what SERCA does is, I'm just going to put a pointer on. SERCA is a really important pump where it can draw calcium back into this part of the cell. Now, calcium is required for contraction. So normally calcium comes into the cell, helps the muscle contract, but then we need to remove the calcium so that the heart can then relax. And so what SERCA does is it draws this calcium back out of the cell and helps the heart relax. So if we have less of this protein, then you can imagine that the heart is not going to be able to relax as efficiently. And so from these findings and all the data that we found from that big resource paper, we have a few kind of 3 areas that we're following up. So the first is doing something known as spatial transcriptomics. Now, this is real cutting edge technology that has really only started to evolve over the last few years and be used a lot in molecular studies. And ultimately what this lets us do is to really hone in on what genes are being switched on and off in the heart, but at a specific cell level. So the project we did, we took a whole bit of heart tissue and we looked at this as a whole, but I'm sure you can appreciate that our heart is made up of lots of different types of cells that all have different functions. And so by doing something like spatial transcriptomics, we're really able to actually hone in on what's changing at a cell-specific level, which will really help for future kind of treatment and preventative medicine. We also currently have a study that's being done by one of our PhD students on actually looking at the left ventricle versus the right ventricle with aging. So our heart has 4 chambers, and 2 of the most important are the left ventricle and the right ventricle, as seen here. And so my study focused on looking at the left ventricle. So we specifically had bits of left ventricular tissue, and that's because this is kind of, it's the part of the heart that pumps blood out to the body, and it's also the part of the heart where we see disease affect the most. So most of the time when we're treating heart failure and things like that, we're treating changes that have happened to the left ventricle. And so it would be really interesting to understand, why do we see most of these changes at the left ventricle and not the right ventricle? So we have a really interesting study going on at the moment where we've now got samples from the same people, right ventricle versus left ventricle, and over the age span as well. So we've already started to see some really cool results with that. And finally, kind of the third arm, that this is more on the future horizon, is to really understand what's happening with that protein SERCA. And so because we get these biopsies that Sean was referring to earlier that we currently collect from patients undergoing surgery, most of these patients have a deficiency with that ability to relax of the heart. And so if we could take some of that tissue and isolate cells, so cardiomyocytes, which are the contractile cells of the heart, we increase SERCA expression and see if this can improve function. And so ultimately this would be kind of experiments that are really needed to translate anything into the clinic. So this is definitely something we're planning for in the future. Okay, so to move on now to kind of the second arm of my work, which is now looking at aging of the human aorta. And so again, why is this important? So just like the left ventricle and the heart, how the aorta changes at a molecular level in humans is really not well characterized. And we know that this knowledge will help us understand why the vessel becomes stiffer and more disease-prone with age. So we know things like hypertension and aneurysm formation significantly increases in older individuals. By understanding these molecular kind of events that happen with aging, this will really help us guide development of targeted therapies and early interventions that might help us extend healthy aging rather than then flipping to kind of disease. It's also important to note the connection between the aorta and the heart as well, and why looking at both of these is so important. So the left ventricle of the heart pumps, as I mentioned, pumps blood out to the rest of the body through the aorta. So they're actually connected structurally as well. So the left ventricle is directly connected to the aorta. And we know that as we age, the aorta becomes a lot stiffer. So our aorta is ultimately an elastic tube. It needs to be able to expand and recoil nicely as high pressures of blood are moving through it. So if this tube becomes stiffer, the way that blood moves through it changes dramatically, and our left ventricle then has to work a lot harder to be pumping blood out into a stiffer tube. So this is ultimately how the aorta changes, ultimately will also likely have an effect on how our left ventricle is working as well and have effects on the heart. So I think understanding how both change with age is really important in understanding disease, aging diseases. So we have a really unique study design, which due to the samples we have in the heart bank has been possible. So we have about 15 healthy aortas that we have in the Sydney Heart Bank. And we also have left ventricles from majority of these patients, I think -1. So we're actually able to look and do a study of the aorta and the LV from the same people and see how they change with age. We have a really nice age range. So the youngest is 10, ranging all the way up to 65. So this really helps us look at the whole kind of lifespan. The first thing we kind of had to do was to make sure all the aortas were healthy. So we did some basic H&E saying just to confirm, that they're healthy. And they all were, and we have some really nice pretty images that we'll be able to use. And then we took these samples and we looked at the protein levels like we did with the heart and compared this with age. So how did these proteins change over time with age? And were there any that significantly changed? And so from our initial analysis, we have seen that there are quite a few proteins that do change with age. So this graph here, all the dots are showing an individual protein, and any on this side are showing proteins that increase with age. And on the right-hand side, any that are going up with age. And so you can see there are a number of proteins that actually change with age. And when we look at some specific ones, we see these really, really clear patterns of some that are decreasing, most of them decreasing with age. So we can see here on this axis is age, and on the y-axis is the actual amount of that protein. And we see that there's this really clear linear relationship with age of a lot of these proteins. Interestingly, most of these proteins, their function has to do with the structure of the aorta. And so as I said, as the aorta, we know it stiffens as we age. If we have these decreased or changes in proteins that are really important in how the aorta is structured, that would make sense in seeing changes in how the aorta is actually functioning. So this was a really cool result, and we're further kind of exploring what this might mean for the aorta. Because we also, as I mentioned, uniquely have this matched sample set where we have the aortas and LV from each individual, we're also able to kind of explore whether the changes we're seeing in the aorta also happen in the LV as we age, or is it something that's happening specifically in one of the tissue types? And we found a mix of both. So we have found proteins that actually change at the same way with age. So for example, these two proteins here both increase in the aorta, which is the blue line, and the LV is the orange line. So we could say that these proteins are likely proteins that have age-specific roles and change with aging. And it's actually seen in other literature that these type of proteins change in other tissues as well with aging. So this is kind of a result that did make a bit of sense. Interestingly, though, we did see a number of proteins that change very specifically in the aorta. So for example, we can see this protein here, tubulin, decreases with age in the aorta, but stays quite stable in the left ventricle. And we see this for a few proteins as well. So this is highlighting that aging changes can be very specific to the actual organ we're looking at. And this will really help us then if we want to guide targeted therapies, that it's not a one size fits all kind of idea. So our aortic work is that we've got actually more data that's actually currently being analysed. So we've also performed something called single nucleus RNA-seq, and this is actually looking at how the genes are expressed. So all the data I've shown previously is looking at the protein. This is looking at how the genes are expressed in individual cells. So similar to the spatial transcriptomics I was talking about, this actually allows us to look at individual cell types. So here on this graph, every color is a different cell type, and we can look specifically at how gene expression is changing within each cell type, which again will give us a better idea of structurally what might be happening in the aorta. We also have started to undertake microscopy, so again, taking pretty pictures to validate some of those results that I was talking about of those key proteins that change. And so this is an example of an image. So our aorta has a endothelial layer, so this inner layer, which is where the blood would be running through here, and a middle layer. And we can see that our protein of interest, for example, here, staining in our smooth muscle cells. And we were able to then, we can look at younger and older and again compare the amount of these proteins between the groups to really validate and try and understand a bit better what's going on with these proteins that we're seeing change. And so that's kind of the update where we're at with both projects. And obviously this work does not happen without a lot of people getting involved. So we've obviously had a lot of collaborators that have helped us get our cardiac paper published. So we had a lot of collaborators in Melbourne as well as at the University of Sydney. Obviously everyone in the lab working forward, everyone at the Baird Institute. Getting this precious tissue is essential to all of our research, and of course the patients who donate tissue, but of course all the supporters of the Baird Institute really make this research possible. So we definitely want to say a big thank you to everyone who's been able to help in any way.
Thanks so much, Cassie. That was obviously, as always, very fascinating, the work that's being done. I think we've got Paul is in. Let me just double check. Paul, there you are.
Yeah. Hi, everybody.
Yes.
Sorry, I wasn't here at the beginning. Just got caught up. Thanks, Cass. I came in at the end of Sean as well. I have to start by saying that work is well above my pay grade. It's extraordinary. We've always focused on a lot of the work of the heart muscle after heart attacks, of course, what everybody knows. But the genetic basis or intrinsic capacity of the heart to fail or recover that work has really only been made possible, or in large part by the work of the Sydney Heart Bank. So it's an extraordinary application. And we've always been interested in the fundamentals of aortic aneurysm disease and how to predict complications and how to predict who to operate on and how far we should go, etc. So extraordinary work. But the link between the two is absolutely fascinating. And on one hand, the role that aging plays is obviously of great interest to all of us, but also quite apart from aging, the links of what we might do to the aorta and how that might affect the heart, and what we might do with the heart might affect downstream of the aorta as well. There's a lot of follow-on work and a lot of application that can go from the work that Cass has just been speaking about. So it is incredibly important, and it's probably far more important than we quite yet realise as we know more about the mechanics of how one interacts with the other. So it really is groundbreaking work and stepping out further from the edge than we have done in the past. So I'd have to congratulate Cass and the whole team in working on that. We're absolutely delighted to be able to help, of course. And I'm very happy to answer any questions, but I suspect the smart questions will have to be directed to the smart doctors. That'd be Cass and primarily. You're on mute.
Yes, I just realised that. I don't think anyone sent questions in, is there? There's one from, oh, hold on, Louise has got a hand up, I think. So, Daria, will you unmute Louise Baird to have a word?
Apologies, my camera stopped working, so you can't see me. Can you hear me?
That's okay. We can hear you, Louise, yes.
Louise.
Hello, thank you so much. It's such important research and it's helping us understand why things change when we age. And these healthy hearts that you found all these changes on, the high proteins or the low proteins, were there any clues as to what might be causing it? Were these people who had high blood pressure but no heart disease yet, or those sort of things? Were there any early clues as what might be leading to all these changes?
So the thing that makes, I guess, these hearts extra unique and special is that because they were accepted as a donor heart. So ultimately they were ticked off as, okay, this heart is healthy enough to go into, someone that needs a heart transplant. These patients really don't have any cardiac risk factors. Their BMIs had to be normal, all of those things. Otherwise, they wouldn't have been able to be donors. So that's what makes this really extra special, because they're defined as kind of real healthy hearts. And so we can really then just pull out the idea of aging, right? So, majority of these changes are likely happening because of that they're a lot older than the younger group. And so we know naturally as we age that we see changes in the heart functionally, like I was talking about diastolic dysfunction, that ability for the heart to relax. Our hearts do become stiffer naturally with aging before any disease, happens. So I think it's really cool that we can start seeing changes that are happening with normal aging and trying to understand, okay, well then why in some people do they develop heart failure? For example, with SERCA, is there in some people, is it SERCA then gets to a point where it's so low that we start seeing changes that actually become then, disease forward. So I think it's us really trying to understand where that balance tips as well, kind of between healthy aging and disease. And that's what we're kind of trying to work on now further.
But can I add to that? So for the most part, however, because you're saying that these are so-called healthy hearts accepted as donors. So for the most part, although there are only small numbers in there when you're looking at many thousands, for the most part, we don't think there are overriding cardiovascular risk factors in that. So we think, at least with what we have, we're signing the changes specifically to aging, if indeed you can do that. Is that what you mean?
Yeah,
yes.
I think, and just to add to that, to what Cass said, Louise, it's actually a historic aspect to do with Australia and heart transplantation, because we really had one transplant centre effectively in the beginning, which was at St Vincent's Hospital. And so logistically, before we now have transplant centres all around Australia, but logistically that meant if there was a donor heart, going back in the 90s even, then they had to think about how to get that, obviously to retrieve that heart and do that. And so there were lots of logistical aspects at play. And what it meant was, look, it might only have been 4 or 5 a year, but there were cases where perfectly healthy hearts from someone who had no other cardiovascular risk factors, these hearts did not go into a recipient. And that also relates to the transplanting technique that, nowadays, transplants can occur even if there's a body mismatch. I mean, even people of different, with blood types and someone having hepatitis C. And so it's evolved so much. But back then, particularly say in the 90s, a lot of criteria had to be met in order to transplant someone. So this is why these donor hearts are truly unique all around the world, because there are, of course, other heart banks all around the world that collect diseased hearts. But this collection of donor hearts from 3 months to 65 years of age is the only set in the world where we can truly say there were no confounding factors which would otherwise give you those changes. Whereas today, when a donor heart, in inverted commas, is collected by a heart bank, invariably that donor heart's collected because there were concerns that there was some underlying cardiovascular disease or too many cardiovascular risk factors, and they did not want to take the chance of transplanting that heart into a recipient. But our samples are really unique. And when I think about inquiries from other institutions, as I said, a lot of those big institutions have these heart failure samples, and they have them in big numbers, some of the US institutions, but what they don't have are the comparator groups. So that's why our goal has been to create an atlas that's freely available, that people could look at all of this information, and it becomes a worldwide resource for anyone to look at with cardiovascular aging of the heart and the aorta. And that is our big goal, to create something that you can log in and as a researcher anywhere around the world and look at how does the human heart age, how does the human aorta age, and to make that freely available so that we can really further advance our understanding of cardiovascular aging and how that relates to disease.
Fantastic, thank you.
Anybody else got any questions? If you do, you can just click on that raise hand there, and I'll know that you want to ask a question. At the very top, there's a little hand that says raise. Yeah, that's Laurel. I'll let you in, Laurel. Hold on a sec. One sec. Can you do the mic, Daria, for Laurel?
Yep, Laurel, you can unmute your mic now.
At the top of the screen, there's a little mic. It says camera and then mic, Laurel, and then you can unmute. We can't do it, Daria.
Yeah, it says
There it
mute.
is.
Can you hear me?
Yes, we can.
Yeah, right. I just want to know, how much control do we have over aging? You know, I have a history of heart problems in my family. Now, I'm very careful with my diet, make sure I exercise, do all the right things. But is it actually in the genes? How much control do I have over it personally? You know, have you studied that, or is that impossible? Or
no, it's a great question. I guess in one respect, that's the basis of what we're studying, because here we will just look at the natural changes of aging at that molecular and cellular level and see what actually just happens as we age and what changes lend themselves to disease. So there can be changes, for example, in the aorta that lead to more stiffening, or in the heart where the heart's not able to relax as easily. And we know that there are diseases that actually have these same features, but at a higher magnitude. So we're really able to understand that. And if we can understand how those natural changes of aging, if extrapolated, could then lend itself towards disease, then of course we could look at targeted therapies that may slow that down. But to answer your question regarding what you could do now from a clinical perspective, I guess there's always this very interesting relationship between our genetics, how that changes with age, and then environmental influences. And what we know, I guess what I talk to patients about if we're talking about cardiovascular disease, I say it's like there's a bar. And if you go above that bar, you develop cardiovascular disease. The question is, where did you start below the bar? Somebody with a strong family history may only be a short way away from that bar, whereas somebody with no family history might be quite lower down. And then it's simply an accumulation of risk factors until you go over that bar. So in patients, for example, who have a strong family history of cardiovascular disease, straight off the bat, we would aim for lower levels of cholesterol, blood pressure, weight management, prescribe more exercise, really be more diligent. There's little leeway, if you like, and you would be more stringent from the get-go. And you try to do that from a young age to prevent cumulative effects of those risk factors. That's not to say that somebody with no family history or that we would give them free rein, but there's an interesting aspect when you look at, say, cholesterol, and the notion of saying a cholesterol is normal or not just based on a pathology blood test is out the window today. We really look at an individual's risk and we determine if their cholesterol, for example, is appropriate for that risk. And likewise for blood pressure, blood sugar levels, weight, all of those other aspects. Mm-hmm. So I think, you know, if there is a family history there, we'd certainly aim for more stringent lifestyle measures and targets to reduce the risk of cardiovascular disease.
So really good luck, bad luck, I suppose, in a way, you know. I just have to be very careful.
But in some ways, people who have the family history can be more diligent and take those means to prevent disease than sometimes people without that family history or knowledge may not be so much, you know, and so I think it's knowledge is power, and having that knowledge means you can be more diligent and reduce your risk. That's the other aspect. The studies have consistently shown when people are deemed to be of a higher cardiovascular risk, by whatever means you use to work that out, including these risk calculators, you can lower someone back to low risk by being more stringent with their risk factors. So it does work to lower your risk.
Okay, thank you.
Thanks, Laurel. Anybody else? We had lots of people register today, but not many able to obviously attend. Middle of the day, sometimes a bit hard. So they'll all get a recording, as will all of you, if you wanted to go back and listen again. But if anyone else had a question, maybe, or are we all done? You could just raise your hand up if you did, but no. That's all good. Well, thank you so much for coming along today. Thanks to Cassie and Sean for your presentation. Thanks to Daria for helping me technically. And thanks to Paul for coming along as well in the end. But thanks for your support as always. I think all of you here are long-standing donors. None of you are new donors, seeing all your names. And we really do appreciate your support. It makes a lot of our work possible. So thank you very much, and we'll see you at the next webinar.
Thank you.
Bye-bye.
Thanks,
Bye-bye.
everyone.